Bismillahirrahmanirrahim. In the name of Allah, the most gracious, the most. Nauman Khan from Department of Botany, Islamic Ali Peshawar, Pakistan. This project is jointly collaborated with Department of Crop and Animal Production, Sasson Vocational School, Batman University, Batman, Turkey, with paper ID 664 with Professor Dr. Elikan Kaplan. Title of my presentation is Influence of Adaptive Factors on Vegetation and Plant Community Dynamics in the Dry Temperate Region of Chitral, Pakistan. These are contents of my presentation. Next, introduction. Diversity of vascular plants. So plants are critical for driving biological processes, improving productivity, and establishing numerous terrestrial habitats. Furthermore, plants are a basic and important components of biological diversity. Biodiversity refers to the variety of living beings at all levels of organization, ranging from genes to species to habitats and ecosystem. It refers variety and variability of plants, animal and microbes within an ecosystem. Vascular plant biodiversity worldwide. So to tell that 3,08,312 vascular taxa were reported globally, in which angiosperm were 2,95,383, gymnosperm were 1,079, ferns were 10,560 species, and lycopods were 1,290 species, which are currently being described globally, in which 400 taxa, including 169 genera, 45 families, are endemic to Pakistan. Geographical region of Pakistan. So Pakistan is divided into four different geographical region. First is Irano-Turanian region, which make 45% of the total Pakistani flora. Second, Sino-Himalayan, which contribute only 10% of the Pakistani flora. Third, Saharo-Sindhian, which share 9.5% flora of Pakistan. And last, the fourth one, Indian element, which constitute 6% of the Pakistani flora. Even though the Saharo-Sindhian region has the largest area, it has the least variety of species among all geographical regions. Vegetation dynamic. So vegetation may be defined as the flora and fauna of an area, including all known taxonomic groups and their physiognomy. Physiognomy refers to the external appearance of plants, whereas floristic composition refers to the types of plants present in a community. Community morphology and structure describe the physical characteristics and arrangement of plants within a community. This is map of the study area, Ujor Valley. You can see figure A. So figure A is the map of Khyber Pakhtunkhwa province. B is the district Upper and Lower Chitral, and C is the map of the research area, Ujor Valley, and their different sites. For example, Las Susum, Petagram, Susum, Brishgram, Luligram, Gali, Hinjil, Kirchum, Sunij, etc. etc. Highest peaks of the Ujor Valley. So Susum were the highest spots, contributing 11,221 feet above the sea level, followed by Kiar, which is 10,554, while the least lowest peak of the valley is 6,500, which is Gali. Aims and objectives: to investigate how adaptive factors influence the composition, distribution, and dynamic of vegetation and plant communities in the dry temperate region of Chitral, Pakistan. So there are three objectives in my study. First, to identify and classify plant communities present in the dry temperate region of Chitral based on floristic composition and abundance. Second objective is to analyze the physicochemical properties of soil associated with different vegetation types. And the third one is to assess the relationship between soil characteristics and plant species distribution using statistical and ecological modeling approach. Material methods. Methodology. So this is my methodology of the study. I conducted a field survey in between 2021 and 2023. So I selected 22 spots from which I collected different plant species Then I preserved all the plant specimen, and then identification were done with the help of Flora of Pakistan, Tropicos. Grasses were identified with the methodology of Ahmed and Stewart. Gymnosperm were identified with Chaudhary. Pterophytes from the flora of North America and flora of China. And lastly, vouchering were done, and then herbarium deposition. Data analysis. So, two methods were applied in my study: transit and quadrant method for vegetation sampling using the standard protocol of Seltzer and Welling by 2004. So all the coordinates were systematically placed along each transect at 200-meter interval using a GPS. Coordinate of 20 into 20 meter for trees were selected, 10 by 10 meter for shrubs, and 2 by 2 meter for herb. Quantitative analysis of vegetation. So the following formula were used to compute the quantitative properties of the vegetation, which is stated previously by other workers. Barnett et al. 2018, Anwar et al. 2019. So all these formulas, equation were given for quantitative analysis of vegetation: density, relative density, frequency, and relative frequency of citation. Cover, relative cover, and importance value index. Diversity indices, species per species richness. The following formula were analyzed: Shannon index, Simpson diversity index, Pool evenness index. So all these formula was applied. Cluster analysis and two-way cluster analysis were also applied, which is previously reported by Ahmed et al. 2023. The classification of plant species and station coordinates into potential plant communities based on their similarity and differences was conducted using PCR software version 5. Canonical software version 4.5 was employed for canonical corresponding analysis to explore the relationship between vegetation and environmental variables. Person correlation analysis was performed to identify the correlation among environmental variables using R Studio. Results: vegetation structure. So I established 4 different plant communities from 4 different spots. The first spot is Gali Hinjil Dronil, in which altitude range 6,500 to 7,800. The soil were sandy loamy, and the dominant family were Selectatusperma crataegus sungerica, Rheumatibeticum community. Side 2 were Brishgram, having altitude range of 8,000 to 9,000. The soil were sandy loamy, and the dominant communities were Populus alpa, Hypopyramnide, and Arenaria gipti community. Side 3 were Susum 1, in which altitudinal range from 9,000 to 10,500. The soil were sandy loamy, and the dominant community were Anobricus cornuta and Coccinia pineticola community. And the last spot were Susum 2, which ranges from 11,000 to 14,000. The soil were sandy loamy, and the dominant community were Berberis calabatris and Ranunculus hortiolus community. This is the clustered dendrogram of 40 station on the basis of Sorensen-Mayer. You can see in different colors. These are 4 different communities which are associated with each other, and they make different groups, different clusters. Two-way cluster analysis dendrogram of 133 associated plant species and 40 station based on Sorensen-Mayer. The black block show the presence of species, while the white color block shows the absence of species. So the black block species were closely associated with each other, and they make different plant communities in different stations. These are the diversity indices of the 4 communities in which species richness were highest in community 1. Shannon index were highest in community 1. Again, Simpson index were highest in community 1. And community 3, there were highest symptom index of diversity. In community 1, polo evenness index were the highest. This is DCA plot showing distribution of plant species in the region, in which Galuna, Chatralica, Acacia monorum, Androsia ojorensis, Androsia stentoni, Astragalus tupinianus, Ranunculus hortulus. These are closely associated with each other and they make one plant community. Also, this has positively strong association with each other. While at the bottom, the left bottom, Rosaicea, Hippopyrum riot, Populus alba, these are far away from the uh, community number 2. This is the community association among each other, which is shown in different colors. So community 1, community 2, community 3 and community 4. Community 1 and community 2 has closely associated with each other, but community 4 are far away from community 1 and community 2. Uh, Simultaneously, community 3 and community 4 were a little bit closely related to each other, but they are far away from community 1 and community 2. CCA, this is CCA plot of the plant species in relation with soil variables. So you can see potassium and pH level. So all these species are influenced by high potassium level and pH level. For example, Androsia stentoni, Juncus squamata, Elagia rhombidia, Dreva pakistanica, which are high tolerated to potassium and pH level, while phosphorus, nitrogen, and organic matter, which are influenced by Ephedra gerardiana, Rosa alpestris, Colutia paulina, Arenaria grifti, etc., are influenced by high level of phosphorus, nitrogen, and organic matter, while calcium carbonate and electric conductivity were influenced by all these species. Again, this is plant communities in relation to soil variable. So community 4 is highly influenced by potassium and pH level. Community 3 is highly influenced by phosphorus and organic matter level. Community 1 is highly influenced by calcium carbonate and electric conductivity level, and community 2 were negatively affected by phosphorus and organic matter. This is correlation among the soil variables. Calcium carbonate. So calcium carbonate is highly influenced by organic matter, and they are less tolerable to electric conductivity and pH. Conclusion: the study documented 133 plant species into broadly 4 plant communities in Jor Valley, Chitral, Pakistan. 4 plant communities were identified along altitudinal and environmental gradients. DCA and CCA analysis highlighted species distribution in relation to environmental factors. Organic matter and nitrogen showed a strong positive correlation, while pH and EC had a strong negative correlation. The study showed that electrical conductivity and calcium carbonate, organic matter, pH, phosphorus, and potassium were the main edaphic variable that contribute to the formation of plant communities. Thanks to Almighty Allah, the kind, the merciful, who gave me the ability and strength to complete this project, and all respect are for his last, for our last Prophet Muhammad sallallahu alaihi wasallam, who shows me the right path in every walk of my life. A huge thanks to Professor Alakan Kaplan, Batman University, Batman, Turkey, for their inspiring and motivational report. Thank you all.